Hi everyone, today I will be talking about the effects of nitrate on beef cattle performance and greenhouse gas emissions. I'm very excited to talk about this topic because I'm going to show some of my work related with nitrate during my master's degree at Texas Tech University. So to begin with, I'm going to show the total US greenhouse gas emissions. So as you can observe on this figure, most of the greenhouse gas emissions come from the transportation, electricity generation, and industry sector. And only about 10% of the greenhouse gas emissions in the United States only comes from actually the agriculture sector. And whenever we compare the beef cattle, it only accounts for 2 to 3% of the total greenhouse gas emissions when compared to the other sectors. So why do we care about methane then if, as I showed you in the previous picture, most of the greenhouse gases come from other sectors? We care about methane because methane represents a loss of gross energy intake. And over here we have the ruminant, and the ruminant has this anaerobic sac called the chamber, and it produces volatile fatty acids, which is the main source of energy for the ruminant. And the three dominant ones are acetate, propionate, and butyrate. And whenever these feedstuffs are degraded or fermented by ruminant microorganisms, they release carbon dioxide, hydrogen, and hydrogen as well. And when that is released, there's a group of microorganisms called methanogens, which are in charge of grabbing that CO2 molecule and hydrogen, bind them together, and release it as methane production through belching or rumination. And why methane is important to the ruminant, as you can observe here, there's hydrogen production. And if that accumulates in the rumen, it can cause acidosis, and it can be detrimental for both the host and the ruminal microorganisms. For the host, dry matter intake can be decreased, as well as performance. And for the ruminal microorganisms, it will be detrimental for some of those organisms that live on that more neutral type of pH. So as I say, methane represents a loss of gross energy intake. Over here we have the molecule of methane, and as you can see, it has carbon. And that carbon that is released to the atmosphere for methane is not utilized then for meat production or milk production. Therefore, it represents a loss of 2 to 12% of gross energy intake, being concentrate-based diets closer to that 2%, and forage-based diets closer to that 12%. And as if you think in terms of profit or investment, it means that producers lose about 2 to 12% of their investment in feed on methane production. Another aspect to consider when it comes to greenhouse gas emissions are excreta emissions, which I'm talking about manure as well as urine. Greenhouse gases such as primarily nitrous oxide, methane, and CO2 are produced from excreta through microbial decomposition of the organic matter from this material. Feed production, growth of cattle, and management and animal excreta contributes about 8 to 18% of the total greenhouse gas emissions whenever we consider all the beef cattle sector as a whole, which it actually increases that 2 or 3% that I already spoke about. And something important to consider is that about 25% of the total greenhouse gas emissions from beef productions comes from manuring pens and housing. And for me, this is very representative in my perspective. However, the industry and the scientific community have looked for different methane mitigation options, such as nitrates, ionophores, tannins, oils, and feeding strategies. And today I'll be talking about nitrate and how it works and some of the results from my experiment. So how nitrate decreases methane emissions? Over here we have nitrate, and when this nitrate is totally reduced in the rumen, it acts as a hydrogen sink, meaning that there's less hydrogen available for methane production. So that's how it decreases methane emissions. And also in its intermediate state, nitrite, it inhibits methyl coenzyme and reductase, which is a crucial enzyme for the cycle of methanogenesis or to form methane. So the first experiment I'm going to talk about is the effects of calcium ammonium nitrate on performance and methane production of beef steers grazing a mixed winter forage. For this experiment, we had a total of 36 steers. They were grazing a mixed winter pasture, and we had 3 different treatments, which all the treatments were supplemented with 0.3% of the body weight with corn, which kind of represents about a kilogram of corn. And we have PC, which is pasture plus corn, PCN, which is pasture, corn, and nitrate, and PCU, which is pasture, corn, and urea. And these 2 treatments, they were isonitrogenous, meaning that they had the same amount of nitrogen. So this is how we look in the pastures. We had 4 steers that were corn, nitrate, and urea, and that was replicated in 3 different blocks. So here are the paddocks, and as you can observe here, we have the pastures and the TTU beef unit because these animals were supplemented individually daily. And just to have an idea of the amount of work and everything that we did, those animals were walking about 2 miles per day in order to be penned. So what we did is that we brought the steers from the pastures. They were penned individually and then supplemented. And then we waited an hour for them to consume the supplement. And then we took them back to the paddocks. And then after that, we collected the orts and we measured supplement intake. And right now I'm jumping all the way to the results, in which over here we have supplement intake. We have corn, nitrate, and urea. And this is during the grazing period, and this is during the methane period. As you can observe, during the grazing period, nitrate was the supplement that was consumed the least when compared to the other 2 treatments. However, during the methane period, we didn't have any differences, and this will influence on the results from this experiment in another one that I'm going to present as well. We didn't observe any differences in methane production, and we attribute this fact to the lack of consuming the supplement during the methane period. Just for you guys to have an idea, only about 30% of the supplement was consumed during that period. Therefore, we attribute the lack of significant differences to that aspect. And when it came to body weight, we didn't observe any differences as well. Therefore, we concluded that the amount of nitrate consumed was not enough to reduce methane production. And also, if nitrate is going to be used as a methane mitigator strategy, it should be delivered in a different way, because with the corn, it was quite clear that it wasn't consumed very well. And also, nitrate did not negatively affect performance of beef steers. So then what we did is that during that methane period, we also collected manure from each one of the blocks, and we ended up with 9 composites, just as you can observe on this picture. And what we did is that we put 1 kilogram of manure as is inside some static colors. Over here, there's a picture of my advisor and another one of my advisors as well. And we put that manure in those chambers. Then we collected gas samples with a syringe, put them in serum bottles, and then we brought them to the lab and analyzed them for concentration of nitrous oxide, methane, and carbon dioxide. And this is how we look in the paddock. We had 3 different blocks, and each one of the blocks had 3 composites with the 3 treatments. So as I mentioned before, supplement intake was not different during the methane period. And this will affect my results during the excretion emissions as well. We did not observe any differences on nitrous oxide emissions from manure, as well as methane emissions. Therefore, treatment did not have an effect on cumulative. And also the estimated annual manure emissions, this is something that I really like from this experiment, is that they were lesser when compared to the IPCC. The IPCC is the Intergovernmental Panel of Climate Change. We observed that our manure emissions were ranging, with considering methane, between 39 to 63 grams, and the IPCC reports 1,496 versus nitrous oxide. We observed that same effect as well. And this type of experiments actually really help, because they help you have a broader idea of how excrete emissions work on a grazing setting. And the IPCC, most of the values they report come from actually confined spaces. Therefore, we concluded from this experiment that the amount of encapsulated nitrate by beef steers grazing a mixed winter forage did not alter emissions of nitrous oxide, methane, or carbon dioxide. I hope you enjoyed the presentation, and thank you very much.